Thank you for coming. I'm an analyst in the search evaluation group here at Google. I've been here for about 6 months. And before that, I was working on systems biology in my PhD. And a few people from my team suggested giving a small talk to describe what I was doing before coming to Google. And I decided to sign up for a tech talk. So hopefully you'll find it interesting. And if you have similar interests, please talk to me afterwards too. I'm very curious to find out who here has worked on similar stuff. And with that, let's please get the recording started. So the types of systems that I have been interested in are cellular systems that perform very logical functions, mostly using networks, very complex networks of genes and proteins inside of cells. And to give you a few examples, I have 2 videos here. The video on the left, I also have to speak here on the microphone just because of the recording, sorry. But the video on the left here shows a neutrophil in human body, and you will see it chase down a foreign invader, a bacteria. It's surrounded by red blood cells, and the remarkable thing is that it's continuously sensing its environment and is able to follow the bacteria very, very well until it eventually catches it and destroys it, which is a good thing for us. And this video was taken in the 50s, which I find pretty amazing too. So there you go, we got the bacteria. And then the video on the right shows another type of bacteria, E. coli, and they tend to perform a biased random walk in space to on average find the regions where they have the highest concentration of nutrients. And the way they do that is that they have a bunch of filaments that you can see here. So here's one cell, this is the cell body, and these are the filaments. And when the filaments turn in one direction, the cells move forward. And when they turn in the other direction, the cells tumble and choose a completely random direction to continue moving in. But the cell has a way to tune the frequency of the tumblings with respect to the forward motion to, on average, again, find the region in space that's best for them. So here you'll see them do that a bit. The cell here is stuck, but the others are moving. And these videos make clear, I hope, that cells really are dynamical systems that are continuously sensing information from their environment. They're processing it in very sensible ways and responding to it by regulating important functions such as cell movement in the videos that you saw, but also growth rate and important physiological variables like temperature, pressure gradients, and so on. And the goals of this field of systems biology is to really understand the system behavior based on the components that we know exist and make up the system, and to eventually engineer these systems to perform functions that we want them to do. In the last few decades, biology has been accumulating a vast amount of data. So for the important systems, either in humans or in other organisms, we know many of the proteins that are involved, some of the connections between them. But still, there's no system way to bring all the knowledge that we have and really be able to predict what the system actually does and what function it serves or how to modify it to make it do something else. These systems are very complex. So the graph here on the right shows a protein network for a given organism. So each node corresponds to one protein, and there's thousands of them. And all the links correspond to known interactions between them. But the data is noisy. It's typically very limited. There's no time series or anything. It's mostly steady state data. And additionally, there's many levels of organization, starting from the molecular level, cellular level, the tissue level, the organ level, and the organism level. And we don't have a good way to bridge all these hierarchies at this point. And there's clearly a very tremendous impact for being able to understand and manipulate these systems, not only to learn about fundamental biological processes like the cell cycle, in which all of our cells grow and divide in a good way, for example, without copying more DNA than they should, and things like that. And this will hopefully lead to many breakthroughs in human treatments, either by I don't know, gene therapy, or even engineering biosensors, or generating organisms that will help us process some resources, like cellulose, into fuels and other useful chemicals. And there's people right now working on these types of things, so it's a very hot topic at the moment. This is an example of some goats that were engineered by Genzyme to manufacture some drug. You can find them online. So again, there's very few examples at this point where we have been able to successfully engineer biological systems to perform very useful things, and we're still at a very early phase in this journey. And at this point, the field is extremely interdisciplinary. There's biologists, obviously, there's physicists, there's some engineers, mathematicians, and it's really unclear at this point which of the approaches is going to yield insights into how these systems work and how to manipulate them. My background is in electrical engineering and applied mathematics, and of course I believe that some of the tools we use there will hopefully contribute something to understanding these biological systems. And in this talk, I'll tell you about some of my experiences in an experimental project where we studied a system in yeast, and also some more mathematical tools that we've developed to try to work with these systems and understand the different phenomena better. I'll also conclude with some future research directions that people are taking at the moment. So, budding yeast is important to study because in many cases, well, it's very easy to handle, yet it is remarkably similar to human cells in many ways. So the system that I studied, for example, has one protein, the main protein in the system, which is very similar to protein P53 in humans, which is known to be involved in cancer and the cell cycle. So the hope is that by studying simpler systems, we can actually learn about ourselves and come up with insights for healthcare and a bunch of other things. So yeast cells are about 5 to 10 microns in diameter, and they divide almost every 90 minutes, which is nice. They're also very friendly to us. We use them to make beer, wine, and a bunch of other things. So the system that I studied regulates the pressure gradient across the cell membrane. It turns out that the cell membrane of a yeast cell is semi-permeable, meaning that small molecules can go through passively through diffusion, but larger molecules called osmoles cannot. This generates concentration of osmoles inside of the cell, and there's another concentration outside of the cell, which results in a pressure gradient across the membrane, which normally drives water flow into the cell because the internal concentration is typically higher. So this causes the cell membrane to stretch. That's why the cells are round and bulky. And failure to regulate this gradient pretty much completely disrupts the cell cycle, and the cells cannot grow unless this pressure gradient is intact. Unfortunately for G cells, in comparison to the other cells that I showed you at the beginning, they can't move, yet they're subjected to external changes in osmolarity. Therefore, they must have a system to regulate the internal osmolarity. And it turns out they do this by what's called the HOG1 signaling pathway. I'll show you a diagram in a little while. And eventually, so the input signal is the external osmolarity, and eventually the system results in adjustments in the glycerol production, which is a molecule that the cells use to regulate the internal osmolarity. So the initial goals of this project was to first understand the system behavior, and then to try to understand what parts of the system architecture are responsible for its main function. And hopefully that will become clear. So this is a diagram of the system. It's a simplified diagram already. So, and this is typical of many biological systems of interest. They have, again, many proteins that are involved. The input signal is here, osmotic stress, so external concentration of osmolyte. And this leads to a sequence of chemical reactions involving many proteins, eventually leading to this protein called HOG1 going from the cytoplasm into the nucleus of the cell. In the cell nucleus, this protein HOG1 interacts with many other proteins, which eventually lead to the new production of enzymes that regulate glycerol production. And standard approaches in the field gather all the knowledge about the system, and they write down differential equations, nonlinear differential equations, for the concentrations of each of the components. So even for this tiny module here, the authors of this paper, a pretty good paper, ended up with all these many equations. Now the problem with this is that the data is very noisy and is very limited. So estimating a vast number of parameters based on the data is very tricky. And also when you put all the equations together, it's also very hard to see what the function of the system is. Sorry, go ahead. You'll see how you get data for that. It's very tricky. Normally, measurements tend to be steady state and averages across the whole population. What we've done is develop some technique to actually get single cell time series by monitoring the localization of this protein, pretty much, but you'll see that. So yeah, instead of doing this approach, which is again typical in the field, we decided to take more of an electrical engineering approach, where you have a complicated circuit, you grab an oscilloscope, you feed a bunch of different time-varying inputs, you measure the time-varying outputs, and based on that, you get some rough idea of what the system is doing. So in this case, we chose the input, it's clear. We chose a few signals that we thought were meaningful outputs, so the amount of glycerol, the localization of this protein HOG1, and the volume of the cell, which is a proxy for the pressure gradient across the membrane. And this is a more detailed biological diagram, well, an abstracted diagram compared to the previous one I showed you. And this is all really you need to know for this talk. So essentially, you have an external pressure that's a linear function of the concentration of osmoles outside of the cell. You have an internal pressure, and there's a protein in the cell membrane that senses changes in this pressure gradient, and in response to that starts a sequence of chemical reactions known as phosphorylation events. And the last protein in this cascade of information flow gets 2 phosphate groups added, and when that happens, it goes from the cytoplasm into the nucleus, where it regulates an increased production of glycerol. And additionally, there are HOG1 independent factors, like channels in the cell membrane that open and close in response to changes of the pressure gradient, that eventually also lead to the accumulation of internal glycerol. So drawing an abstracted engineering diagram of the same process looks as follows. You have the system input U of t, and then you have a protein in the membrane that measures the difference. That gets fed into the cell signaling box, which corresponds to the cascade here, resulting in phosphorylated HOG. And then HOG and the HOG-independent mechanisms both serve as inputs to glycerol production. And this is what closes the feedback loop. So you can see here that there's at least 2 very basic negative feedback loops, one that goes through HOG1 and the other one that is independent of HOG1. Measuring the system is very tricky. What we did here is came up with a fluidic device that has a chamber with fluid from one of 2 types going over cells stuck onto the slide via some chemical. So we have a bunch of cells that are stuck here, and then we have a computer that controls a valve which regulates which of the 2 inputs we're sensing, and we can use that to construct different time-varying inputs such as square waves or just step inputs. In order to measure the cell dynamics, there are a bunch of very neat biological tricks you can do that take forever to make work, but they do work eventually. So we modify the DNA sequence of this protein HOG1 by adding a segment that gets converted into a protein, YFP, the yellow thing here, that emits a signal only when excited at a certain frequency, so without disrupting the protein function. So when you use a microscope and you shine light at this frequency in the microscope, you get a sense for how this protein is localized within the cell. Additionally, to know where the nucleus was, we added another tag to a protein that's found only in the nuclear membrane. And this is the type of data that we get. It's not as exciting as the first 2 videos you saw because I explained that yeast cells can move very well. So, and we had them stuck in the experiment. But this shows a response to a step input of salt. That was the osmol we used for the experiments. And the one on the left is just phase contrast microscopy. Each little circle is a cell. And what you will hopefully see is that the volume decreases very quickly upon addition of salt. And then over the course of the experiment, the cells resume growth again. So there the volume decreased, and now it's growing slowly back again. And the video on the right shows the localization of this protein HOG1. So that's how it looks at the start of the experiment when the cells are happy. And it's pretty much found in the whole cell. And you will see that shortly after salt comes into the system, HOG1 concentrates in a little part of the cell. That's actually the nucleus, which we can see with another image that I'm not showing now. And then eventually, the localization of HOG relaxes to something similar to what we had at the beginning of the experiment before the input was put. So I wrote some scripts to extract quantitative signals from each of the cells in the experiments. We defined first the raw signal, which is the pixel average of the intensity in the nucleus over the pixel average in the intensity of the whole cell. And there's a phenomenon in this, when you use this microscopy, whereby the intensity of the images decreases exponentially with the number of images that you take, because the protein tags that you put get degraded every time you excite them. So in order to correct for this effect, at least to first order, we then actually track relative changes from that. So by taking the ratio, we get rid of this effect to first order. So the plots that you will see are actually percentage changes from the baseline nuclear enrichment of this protein. And the first experiment we did was to subject the system to square waves of different frequencies. And if any of you are familiar with the standard engineering tools from systems theory, linear systems theory, then you'll recognize this is the body plot of the system. And also, if you have a basic background in this, and by just looking at this plot, you can already see many things. You can tell, for example, that if the system is linear and time invariant, which it's not, but just as a thought exercise, a very useful one, you would know that the Laplace transform of its impulse response would have two poles at the frequency where the slope changes here. And you would also know that the system has a single 0 at 0. Why is that interesting? It's interesting for various reasons, but I think the most interesting one is that you can then apply another very useful result, the final value theorem, which tells you what the steady state level will be for an input response of a given magnitude. And that's obtained by simply evaluating this function at s equals 0, at the frequency 0, to get the DC offset, multiplied by the gain of the input, by the magnitude of the input. So because this is proportional to s, then that tells you that the steady state is 0, regardless of the inputs. So it doesn't matter what type of step input you'll have. This would predict that the steady state will always be the same as the initial value. And that's actually a very hard function to get to if you want to construct it. So then we went and measured the actual step response of the system, and here's what we got. So yes, this is the start level. This is the steady state level in response to a step, and they are the same, like the linear analysis predicted. It had no reason of holding because the system is nonlinear, but it still does. The solid line here corresponds to the average overall cells in an experiment. The shaded region corresponds to the mean plus 1 standard deviation. And so it corresponds to the cell-to-cell variability within an experiment. And the jagged traces correspond to 3 cell responses, single cell responses. We can also measure the relative changes in the volume. And essentially, as soon as you add salt, the volume decreases very quickly within seconds. And then over the course of the experiment, it recovers. And it actually crosses the initial level, which means that at this point, the cells have a healthy enough pressure gradient across the membrane that they can grow and divide, like they were at the beginning of the experiment. So this is another plot that tries to summarize the main system behavior. So we start here at rest at a 0 level of volume, that's a baseline level, and a 0 level of Hog localization. And within seconds, in response to a step, the volume drops within 20 seconds, while Hog stays the same. Then for the next 4 minutes or so, Hog localization increases, Hog gets into the nucleus, but the volume stays the same. And then there's a slow transient where Hog and volume recover slowly. And then the cells get to a point where the pressure gradient is good enough that the Hog level is 0 again, and they actually start growing. So we went and repeated this for many concentrations, and these are the behaviors that we obtained for various cell concentrations. And as you can see, the system is very nonlinear. It has a saturation effect here. So as we double the input from 0.2 to 0.4, we certainly don't double the output. And when you go from 0.2 to 0.6, you certainly don't triple the signal. But again, the most interesting feature for me was that the steady state is the same regardless of the input. And that is very important in biology because you have many variables that you want them to respond to external inputs, but to have a constant steady state, like temperature. You want to have a way to sense the temperature in your environment, but you also need to have a way to regulate the temperature so that it stays in a healthy range where your proteins can actually function properly. And it's the same for every basic physiological variable. So this, the process by which this happens is called homeostasis in biology, and it's also referred to as perfect adaptation. And it turns out that control theory and engineering also has a very good way to understand how this can come about. So the volume also looks good. The volume always drops, and then it recovers. And by the end of the experiment, the cells are growing as quickly as they would be. Sure, go ahead.
I noticed there's no ringing. Or is it just that you're measuring it?
Yeah, no, there's no filtering. It's raw data, and yeah, there's no ringing. So even if you look at the single cell traces, there's a lot of jaggedness here, but this is mostly measurement noise. We don't even really think it's true noise from the system. But an important question is how stochastic these systems are, and no one really knows at this point. They are stochastic if you measure small enough volumes, but it's unclear how relevant that is when you get to more macroscopic or even cellular behaviors. So anyway, trying to understand this perfect adaptation, we can go back to another basic result from control theory that tells you that if you have a system, denoted here by the black box, that responds to a set of inputs, U, and for all the signals in that set of inputs, your steady state is independent of the magnitude of the input or of the signal that you chose in the input set, then you necessarily must be able to partition the system into two subsystems with the following connections here. Essentially, you have a subsystem that senses the signal and responds to the signal and generates your measurable output. But you need to have another subsystem that's called the internal model system connected in a feedback loop with the other one that responds only to the output. And importantly, this subsystem has to be able to generate all the signals in the input set. So what that means for the set of signals that include all the step inputs is that the system must have a subsystem that can generate all step inputs, which is an integrator, put in a feedback loop here. So this tells you that the only way to get perfect adaptation in a mathematical system of ordinary differential equations, it even works for a large class of nonlinear systems, is to have an integral feedback loop. Having just a negative feedback loop is not enough. It has to be integral feedback. And this is something that control engineers have understood for a long time, and they use in many electronic devices, or even in the way to regulate temperature in our house. The classical view of control is to have a controller that implements proportional, integral, and differential control. And they regulate different aspects of it. But we've known for many years that to control the steady state, we actually need the integrator part. So this is just a more general result than that, because it applies to a bunch of signal sets. So the result also tells you that it doesn't matter how much you insult the system here, as long as you maintain this topology of the network, and that this system is still able to generate all the signals in your input set, you will still get the perfect adaptation behavior. So we decided to start testing that, and we did that by insulting the cells in many different ways. The first way we tried was to significantly reduce the concentration of this gene called PBS2, which activates HOG1. So if we completely knock out this gene PBS2, then HOG1 never gets activated. The whole feedback loop is broken and we can't measure anything. But what we can do is modify the DNA too, so that the number of proteins gets significantly reduced. And by doing that, we see that the system still perfectly adapts. Now the response, the amplitude is smaller compared to the wild type behavior, and the response takes longer to settle to the steady state, but the steady state behavior is still the same. Then we tried insulting the system in much harsher ways to find out exactly where this integral feedback was happening. And so, by the way, I didn't say how you can achieve an integrator in a cell. So in mathematical terms, an integrator is very easy. You have so a system of differential equations, and you need to have one variable whose rate of change is dependent only on the other variables, but not itself. So the time-dependent form of that variable is simply the time integral of its derivative. And you don't need to do anything special because its derivative doesn't depend on itself. And there's several biological mechanisms that can achieve that, but it's unclear which one we are observing at this point. But going back to trying to break the perfect adaptation, the next thing we did is adding a chemical that prevents any new proteins from being formed in the cell after you add it. It's called cyclohexamide. And it turns out that even after all protein production is completely blocked, the system still perfectly adapts. And this was a new finding for the system because people believe that most of the recovery was due to new protein production. And by doing this, we proved that it's actually due to increased enzymatic activity of the already existing enzymes. You don't need to make any new enzymes. And then a biologist with whom I was working came up with a very neat trick that allowed us to break the feedback loop here in the system. You essentially modify the DNA of the protein again, but in a very different way, such that in the absence of some chemical called PP1, the protein behaves normally, but then when you add this other protein within seconds, it binds to the protein and it prevents the protein HOG1 from interacting with anything else. So it's a very clean modification. Otherwise, you can resort to knockouts, but when you do a knockout, the cells have plenty of time to readjust all the other gene levels. So you have no idea what the resulting topology even of the network is after the cells have time to readjust the levels. Whereas doing this within seconds, we can completely cleanly break this feedback loop. And it turns out that when you do that, the system doesn't perfectly adapt, finally. So that means that by breaking this feedback loop, we were able to bypass the integrator, implying that the integrator, so the remaining feedback loop here doesn't include the integrator. And by some other analysis, you can also show that the integrator is actually downstream of HOG1 but upstream of the glycerol production box. So this already limits the set of molecules that biologists need to look at to figure out where the integration operation is coming from. Then the other thing we did was try to figure out how many integrators the system has. And we can again use the internal model principle for this. So suppose that now instead of considering the set of signals consisting of step inputs, we consider a set of signals consisting of step ramps. You can again apply the internal model principle, and that tells you that the system will only perfectly adapt to those set of signals if it has 2 integrators. Similarly, if the system had 3 integrators, it would adapt to the set of all parabolas. But in any case, we were able to experimentally create some linear-looking thing that saturates at some level. And this is more like a parabola because it was very hard to get it linear. What you see in these experiments is that the Hog1 level and the volume, they don't perfectly adapt. They only do so after you reach the plateau. Then they quickly perfectly adapt. So this shows that the system has effectively just 1 integrator. So not 2 or 3, because that was also the trick. It has exactly 1. And that's pretty much what we found for this system. Do you have any questions up to this point? The thing sort of makes sense or no? Okay. But there's very few engineers in this field, and we were the first ones to actually be able to apply any type of control and engineering principles to an experimental study of a biological system and make a contribution. So I hope that this will also spark more work in other random systems using similar tools from systems. And hopefully we'll get more engineers to participate in this. So at this point, it's mostly physicists and biologists. So now I'll tell you a bit about how to model these systems. That's also a very tricky question. So it's coming from the other end. Suppose that you know all the molecules involved and all the parameters. What types of equations do you write down to understand the dynamics of the systems? And it turns out that there's 2 very important parameters that determine the type of model that you need. There's the mixing rate, mostly diffusion. If you have a system where diffusion is very slow, then you will have spatial gradients in the concentrations arise. So you will need to account for those gradients as a function of space. Similarly, if the system is small, then you need to account for stochastic effects because reactions only occur when 2 molecules diffusing at random collide with a high enough energy to actually react. And at this point, it's unlikely how small a system needs to be for stochastic effects to be important. And it's also unclear how slow diffusion has to be in order to include spatial gradients. And it's very tricky to measure any of these things. So it's very tricky to also just measure it first and then see what you need to do. So most people work at this point in the regime that's here, the simplest one, where you have fast mixing, so no spatial gradients, and the systems are large enough that they're deterministic. But we don't really know that the systems are large enough for it to be deterministic. It's just that this is the simplest modeling framework that we have. And people pretty much just know how to work with this framework. Because even this framework is already kind of complicated. You have a set of coupled nonlinear differential equations and a bunch of parameters. So you can use this simple approach to even start to understand how signal processing can occur within cells. So there's many motifs that are present in many different biological systems, and this is one of them. You have a protein that can be inactive or active, and it switches between the 2 states as a function of the concentrations of 2 enzymes, typically one of which is changing as a function of time, E1. So this is the input. And there's many general tools for this framework. A very useful one is a technique based on singular perturbation theory for separation of timescales. So normally, to model the concentrations of this system, you would need 3 differential equations, nonlinear. But after separation of timescales, you can obtain a reduced model that looks like this for a single variable. And then you can actually analytically understand the behavior of this single equation, because there's many techniques to do that. And then people have found, they have studied this for decades too, and they have found already that at steady state, for example, the system can behave in very different ways. In one case, the system output first doesn't increase at all as a function of the input until you cross a threshold, at which point it completely saturates. So in a sense, this digitizes the signal. And they had also found this other regime that's more hyperbolic. And they were hypothesizing that for analog transmission of signals, the cells probably wanted the system to be in this other regime. It turns out that by looking more carefully at the separation of timescales and at the different possible behaviors, we see that the parameter space, so there's 4 constants, k1, small k1, big k1, and similarly for k2, are pretty evenly divided into 4 regimes. So there's 2 new ones. And in particular, there's a regime that's linear, not only at steady state, but also in the dynamics, unless saturation is reached. And of course, as engineers too, we know that if we want to transmit analog signals faithfully, we want to be in a linear regime. So cells may actually be using this regime to transmit analog signals, not the one here. But again, we don't understand when to include stochastic effects, and the main reason for that is complexity, that the model becomes significantly more complex. So other research directions involve grabbing the complicated stochastic model here and developing some model reduction tools to make it as useful and as tractable as the simpler one that's based on mass action kinetics, which ignores fluctuations. And I did a few projects in this area. So the standard simple model looks like this. You have a state vector of concentrations. So if you have n species in your system, you have n entries in this vector mu. And the derivatives of these concentrations are a function of a matrix S, the stoichiometry matrix, which tells you how much each species changes with each type of reaction that can occur in the system. And then you have a rate of reaction rates, whose entries are nonlinear functions of the state variables. So that's the simple model. When people work with the probabilistic model that includes stochastic effects, then you end up with a discrete state continuous time Markov chain model with the following Kolmogorov equation. So you have an equation that tells you how the joint probability distribution of molecule numbers x, so this is a vector of molecule numbers, changes as a function of time. And you have L different reactions of the system, reaction L, little l, changing the state by SL. So this is the standard Markov chain equation. But the problem is dimensionality here. You have easily tens of variables, each of which can have from 0 to thousands of molecules. So the number of states quickly explodes. And I don't know that even here at Google we will be able to solve it computationally. So people tend to do Monte Carlo. It's very computer intensive. But instead of doing that, because you can't really do any analytical work with Monte Carlo, you just sort of like integrate it and see what you get. We came up with a moment truncation approach. So we derived the exact equations for the evolution of the means and the matrix of variances and covariances in the system. And it turns out that because the probability transition rates in these functions A are nonlinear functions of the x's, the evolution of a moment of degree n of the distribution always depends on higher order moments. So you end up getting an infinite system. You approximate it by truncating it in different ways. So here we did probably the dumbest way of simply assuming that central moments higher than 2 are 0, which would be true, for example, if the underlying distribution would be a multivariate normal. But there's many other approaches that people have tried. And you end up with these very tractable equations for their evolution. This is almost a Lyapunov equation. And people in control theory are very familiar with these equations and know how to work with them very well. It's very easy for computation too, and for numerical integration. And then this is the equation for the mean that we get, where it looks pretty much like the other equation for the simple framework, but it has an additional term which incorporates the effects of the variances and covariances, so fluctuations, in the behavior of the mean. And it turns out that this can make a very significant impact. Also, having the equations in this form helps you very neatly see the connection between the more complicated framework and the simpler one. So omega is the system volume. Suppose that we take the limit as we make the system larger, so this is going to infinity, while keeping the concentrations constant. This is a standard limit to take in statistical mechanics. When you do that, this term goes to 0. And the only terms that remain for the variance equation are the ones here, where m is actually the Jacobian of the deterministic system. It is very often the case that this Jacobian has all its eigenvalues with real parts that are negative. And therefore, when you integrate this equation over time, this converges to 0. So the fluctuations all go to 0 in that limit, in the macroscopic limit. And this term is such, I don't show the formula here, that it also goes to 0 when the matrix of variances and covariances is 0. Therefore, in the large volume limit, you can see very neatly how this system converges to the equations that everyone uses that are macroscopic. And then when you simulate it in a variety of systems, you can see that the behavior you obtain from these approximate equations is much closer to the Monte Carlo behavior. So in this case, the black line corresponds to the solution of the simple system, and the blue dotted line corresponds to the mean of the ensemble obtained from Monte Carlo, with the red lines denoting the ±1 standard deviation. The jagged trace corresponds to a single realization of the stochastic process governed by this Markov chain. And the solid lines correspond to the approximations from these equations. So you can see that it's already much better than the mass action kinetics model. And this is a case in which the behavior differs quantitatively, but not qualitatively. There's other cases where the behavior even changes qualitatively. For example, if you treat the system deterministically, the system doesn't oscillate, it settles to a steady state and that's it. But when you incorporate stochastic effects, you can see that the system oscillates perpetually. You end up getting an oscillator, which are also very useful and very common in biology. So this can make a very big difference. And then we also developed some technique for separation of timescales in the stochastic case. So again, for nonlinear deterministic systems, it is pretty standard at this point to use singular perturbation theory for separation of timescales. But it's only recently, in the last 3 or 4 years, I guess, 6 years, that people started thinking about how to do this for the stochastic model. And they didn't have a good way to identify which variables should be slow and which would be fast. So we came up with a model that does that. I probably won't get into the details, but if you're curious, you can send me an email or you can just look at this paper we have here. And then once you identify slow and fast variables, you do a change of coordinates into those variables, and that results in a very neat form for the stoichiometry matrix, where a bunch of entries are 0, and some of the blocks have full row rank. And what that does for you is that you can then go partition your state space into slow and fast variables, you partition your stoichiometry into slow and fast parts, and you end up with an approximate equation for the joint distribution of only the slow variables. Now again, when you do Monte Carlo of a Markov chain, you need to typically simulate every change of state, right? So the amount of computation is proportional to the number of changes in state over a given course of time. The problem with not doing separation of timescales is that you end up generating random numbers for every reaction that occurs in the system, and most of them are fast. And the larger the separation of timescales is, the larger the percentage of the reactions that you will be generating are fast, and you don't typically care about those. So by getting this approximate model, then you end up only simulating slow reactions. So it's a huge gain in computational cost. Additionally, you can use this equation to obtain approximate equations for the mean concentrations and variances and covariances, and then also do some nice analytical studies on that too. And then you can also obtain an approximate equation for the fast variables conditioned on the slow variables. And by the way, this technique, I don't know if we use it here for PageRank. I'm guessing not, but it could certainly be applied to PageRank. There, the idea is just to cluster the states in the Markov chain in such a way that it's almost in block diagonal form, but there's very small probabilities to transition between clusters. And then you can pretty much apply the same type of method and get an approximate result that will be a lot cheaper to compute. Here's an example of how this looks like. So you have an enzyme, a substrate, forming a complex, resulting in a product and an enzyme. So this is very common in chemistry and biology. And singular perturbation for the macroscopic model resulted in the very famous equation called the Michaelis-Menten rate law that's used throughout chemistry and biology. And when you consider the system stochastically, you end up getting this type of distribution. So this shows the joint distribution of the complex and protein and total substrate, defined as the free substrate plus the complex, and the probability of that, the joint probability of that as a function of time. The two plots are the same. The one on the right just shows the distribution from above, and this one just shows it from an angle. So there's a fast transient that you saw there, followed by a slow transient. And essentially what this separation of timescales method allows you to do is to assume that the fast variables are always at a quasi-steady steady state from a statistical standpoint. And you end up getting an equation for only the slow dynamics, which are a lot easier to handle and deal with. And then you end up with a generalization of the Michaelis-Menten rate law, which normally only incorporates this term for the evolution of the mean concentration of the substrate. And then you end up with a correction that depends on the variance. So this is the stochastic correction. And again, you can combine this slow and fast decomposition and moment truncation to obtain two different models, but the two operations are not commutative, and it's still unclear what's the best order to take when using both of them. So in terms of future directions, there's many things that remain to be done. A very cool one would be to generate synthetic biological circuits by inserting genes of our choice, connected in a negative feedback that implements integral feedback and therefore achieves homeostasis. Another cool project would be to start playing around with also synthetic systems that generate certain types of spatial patterns. Alan Turing, very famous in computer science, was one of the first people, if not the first, to show how a set of coupled nonlinear differential equations with diffusion can give rise to pattern formation at steady state. And that's been used to explain, for example, the pattern formation in zebra skins and in a bunch of processes. It's very important in development as well, biological development of an organism. And it would be very cool to actually generate artificial systems that do this, and to control the parameters that determine the type of shapes that you get. There's many theoretical studies, but it'd be cool to actually do it experimentally. And it'd be also very cool to understand much better how signals get propagated and get processed within the cell. Additionally, it would be also very useful to develop model reduction tools, not only for the stochastic system, but the stochastic system with spatial fluctuations. So this is something I'm interested in pursuing further, probably. And that's all. These are my collaborators for the projects I told you about. Dale is a biologist with whom I worked on the yeast project. Alexander is the head of the lab in which I worked with Dale. George, he's a professor in EE. He was my thesis advisor, along with Leonid, a professor in physics. And these are two collaborators in some of the other projects that I spoke about. Actually, Jay worked with me on the yeast project too, and Remy on the separation of timescales. So yeah, with that, I'll be happy to take any questions if you have any. Go ahead.
So going back to just the biology of the cell, how do you think about isolating the system? It seems like almost every reaction in the cell is changing osmolarity.
The whole change of reactions is what, sorry?
I said almost every reaction in the cell changes osmolarity of the cell, right? So it's kind of weird to think of that you've isolated it to a single.
Yeah, so every reaction in the cell that generates more molecules than it consumes, and molecules that are large enough that they won't go through the cell membrane, will change the internal osmolarity. And we don't, I should probably repeat the question for the video. The question is how to better account for changing internal osmolarity, given that any reaction inside of the cell changes the internal osmolarity, correct? Yeah. So yeah, I don't have a good answer to that question, but it has been measured that in response to changes in external osmolarity, glycerol accounts for about 90% of the increase in internal osmolarity. So I don't know how the cell chose to use that as the main molecule for regulation. It may be that it has vast amount of reserves of whatever makes glycerol that it can quickly break up into more molecules and therefore quickly increase the internal osmolarity. But yes, you're right that other reactions are also contributing to that as well. But having an integral feedback mechanism like the one the cell has would also take care of the steady state. So in a sense, it is robust to those types of things too. Sure. Go ahead.
Can you describe a little more closely how you get an integrator at the chemical level?
Oh yes, I forgot to do that, sorry. Thanks for the reminder. So one way to do it is precisely with this type of motif. So I don't know all the ways in which you can do it, I actually only know one. And no one has been able to find actually molecularly how this happens. And it's a very interesting problem to have. That's why I think it'd be a great idea to generate one synthetically and show that, you know, there's one way at least where you can do it. And the way that I know mathematically can give rise to that is, again, if you have a motif like that, where you have a protein that's responding to some input E1, and that can be in one of two states. So you end up getting these equations for its rate of change. And you can already see here that if you are in a certain regime, so if you tune the parameters so that the amount of total substrate minus active protein, so this here dominates the denominator here, then essentially the first term here will only be equal to K1 times E1, right? And then additionally, if the amount of enzyme plus this rate constant is a lot smaller than the active protein, then the A dominates the denominator here, and it cancels the one in the numerator, so you end up with only K2 times C2 in the numerator. So overall, in that regime, that's one of the 4 regimes here, you end up with an equation that looks like dA dt equals K1 E1 minus K2 E2, which is already an integrator. So the key there is to have essentially a substrate protein that's a lot more abundant than any of the 2 enzymes that modify it, and to also have rate constants, the Michaelis constants, K1 and K2, that are very small to sufficiently small. So you could potentially do that by tuning the levels of the genes. But again, I don't know if the cell actually does this in some system. My guess is yes, but I have no idea. Go ahead. That is also controlled. So by some channels in the membrane, at least. Maybe there's other mechanisms as well. So there are some proteins that form channels across the membrane that open holes in the membrane that are big enough to passively let bigger molecules through. And sometimes they can be very selective. So there's channels that facilitate the flux of glycerol out of the cell. And it turns out that these channels close when the pressure gradients change. So that's another way to regulate the export of glycerol. So it's all very tightly regulated, and also there's many timescales involved. Like in this experiment, we only showed the first hours of the response. It turns out we also measured changes in gene levels resulting from addition of salt, and within an hour or 2, you see many genes whose concentrations increase a lot. And it seems that the cell has different mechanisms to respond to this shock. The first one is using its reserves to produce enough glycerol to restore the gradient. But once it does that, it seems like it also produces more molecules that are involved in the production of glycerol, so that if it gets further shocks, it will be able to respond to them even better.
Your purple line there looks like adding a resistor to lower the Q.
Adding a resistor to what, sorry? Yeah. Yeah.
Is there anything going on there that's similar, or is that just?
We have no idea. It seems that we are, I mean, at the very least, decreasing the overall gain of the feedback loop with the integrator. And that's part of it, but other than that, we don't know. But I do believe that just people who have a lot of experience in signals and systems and in circuits would be able to contribute to decipher the complexity of these systems. The trouble is that the energy barrier is pretty large. You do need to learn biology very well, and you need to invest a lot of time doing it. Even getting this data took like a couple of years. You need to start by genetically engineering your cells, by learning microscopy, creating the fluidic device, optimizing concentrations, everything, doing the software to extract the signals. The software is the easiest part, by the way. Most of the problem is getting relatively noiseless data from the system. So even though this conceptually seems very simple, actually, you know, the reason why people didn't know in this system has been studied for decades too, and they didn't know that, you know, it perfectly adapted. The reason is that they weren't able to measure it to this degree of accuracy before. It's just complicated and takes a long time. It'd be nice if we could actually just plug it into an oscilloscope and then just measure whatever. Okay. Well, thank you for coming.